So we are going to look at sensory receptors and sensory organs and sensory pathways, with emphasis particularly on the receptors of spatial sensations and their related organs and pathways. This is in light of the fact that we have already done the first part of the lecture where we focused on some introductory concepts about sensory systems. And then we also talked about receptors of general sensations. Perhaps if you need to remember what we did last in the previous lecture, we defined what sensation is and we classified them into different modalities. We also define what a sensory receptor is, and we classify them according to their different classification based on the stimulus. We also define what a sensory organ is, and we gave examples. Then in particular, we named the receptors of general sensations, and we stated the sensory modality for each of those receptors. And finally, we described the pathway that general sensations take from the receptors all the way to the cerebral cortex. So that is what we did last time. In this particular lecture, we are going to look at the following. First, maybe we'll review that classification of sensory modalities in brief. We'll also review the structural outline of sensory relay pathways, because in as much as we did it under general sensation, we use the same template even for spatial sensation, albeit some few differences. And so it will be important to have that in mind. Then in particular, we'll focus now on the olfactory sensation. As we do that, we look at the structure of the nose and the structure of the olfactory epithelium as well. We'll also look at gustatory sensation, which is taste. As we do that, we look at the structure of the mouth, the structure of the tongue, the epithelium of the tongue, the receptors of the tongue. We'll also look at the structure of the ear and explain the pathways of hearing as well as equilibrium. And finally, we look at anatomy of the eye, the retina itself, and talk about the visual pathway. While we're there, we'll also talk about the muscles that move the eye and the lacrimal apparatus. Now, I'll do this perhaps in bits, because it's long. We can't do that in one continuation. So maybe the first part, I will handle the first 4 objectives. And that's what I want us to do in this first part. In the second part, we'll talk about objective number 5. And in the third part, we'll talk about objective number 6. I've divided in that way according to the weight of each of the objectives. Let's start with the first objective there, which is basically to review the classification of sensations. Before that, remember some definitions. We define sensation as that state of awareness of a stimulus, and that we classify these sensations as either spatial sensations or general sensations, and I'm going to revisit that shortly. We also defined a sensory receptor, and we say that sensory receptors are structures that detect stimuli, and fundamentally they should be able to convert physical energy that is present in a stimulus, whether mechanical, chemical, thermal, whatever kind of physical energy. A sensory receptor must be able to convert that physical energy into nerve impulses. And then we classify those receptors according to what really stimulates them. So we talked about mechanoreceptors, thermoreceptors, chemoreceptors, photoreceptors, and nociceptors. We also defined what sensory organs are. Say it's an organ that houses a receptor. So things like the skin, the eye, the tongue are sensory organs. So I hope you remember that definition. As with regard to the classification of sensations into general and special, the special sensations are the ones which are housed by the head region. So they're contained within the head region. And so importantly, they are relayed by cranial nerves exclusively. And so these 5 sensations are the ones we are calling special: vision, smell, taste, hearing, and equilibrium of the head. General sensations are not limited to the head region, which means they can be picked even from the rest of the body. And they are also not limited to being relayed by cranial nerves, which means they can also be picked by spinal nerves. There are several general sensations. So that idea of there are only 5 common senses may not be very accurate to this extent. Last time, we looked at the right-hand side of the column, the general sensations. So in this particular lecture series, we are going to focus on this side, the special sensations. And so that brings me to the other thing about sensory relay pathways. We need to review something about the sensory relay pathways. The sensory relay pathway we defined it as a sequence of neuronal fiber processes that carry sensory signals from a sensory receptor all the way to the site of perception in the brain. So that sequence of fiber process of neurons is what you are calling a sensory relay pathway. When you discuss sensory relay pathway, we talk about some entities. The first one is the sensory receptor, which detects the stimulus. After it has detected the stimulus, it will carry that to now the first order sensory neuron, the neurons which are now able to relay the impulses to the central nervous system. Now remember, receptors here could be many varieties. We talked about the receptors of general sensations. There are many with each sensory modality. In this particular lecture, we're going to look at the receptors of special sensations again, according to their sensory modality. Now, the primary sensory neuron, or the first order sensory neuron, is the neuron that is in contact or sometimes part of the sensory receptor. It carries information to the central nervous system. There could be some slight change in this particular lecture, but that's the general outline. First order sensory neuron reaches the central nervous system. So it will terminate either at the level of the brain stem or at the level of the spinal cord. In most cases, therefore, the cell body of the first order sensory neuron will be outside the CNS and will form what we call sensory ganglion. It synapses with the second order sensory neuron. Second order sensory neuron is usually within the central nervous system. This second order sensory neuron is therefore most likely a multipolar neuron. Now, 2 things that the second order sensory neuron must do. One is it must decussate, which is to cross over. So from right to left or from left to right. And 2, second order sensory neuron reaches the thalamus. So as the first order and the second order neurons are traversing the spinal cord and the brainstem, you know, they run in form of groups of axons of neurons. Those groups of axons of neurons with a similar function is what we call tracts. And so these tracts are carrying information to the brain we call them sensory tracts or ascending tracts or sensory pathways. So remember, those pathways are formed largely by axons of either first order or second order neurons as they pass through spinal cord or brainstem. Anyway, the second order neuron synapses with the third order sensory neuron. The third order sensory neuron come from the thalamus all the way to the cerebral cortex. Remember, it will now be on the opposite side because the second order neuron decasseted. The last entity on that pathway is actually the cerebral cortex, and in particular, the region of the cerebral cortex that is involved in perception of that particular sensation. For example, if it is pain, then you know which region of the cerebral cortex is for perception of pain. If it is touch, again, you know that. How about if it's hearing? If it is smell, if it is vision? They have different cortical regions for perception of those sensations. So that's the general outline of how sensations are relayed. These images were in the previous class, and you remember, like in this one, maybe the receptor, there's a free nerve ending. First order neuron enters the CNS. You can see it as unipolar neuron, so the cell body is outside. It's seen as the second order neuron at the level of the spinal cord. In this case, the second order neuron decassettes and climb all the way to the thalamus. As this second order neuron is climbing all the way to the thalamus, several of them will form ascending tracts of the spinal cord and brainstem. It's seen as the third order neuron, which then goes to the cerebral cortex, that region shaded in that color highlighted. And so if you extrapolate that on this one, we notice that actually that corresponds to the postcentral gyrus, the primary somatosensory cortex. Most likely that's a general sensation. Yes, it is. Free nerve ending, so pain, temperature, light touch, they go to that region. This other one showing us Pacinian corpuscle, so maybe the sensation regarding vibration. The first order neuron enters again. In this case, the first order neuron climbed all the way to the brainstem before synapsing. So it will be on the same side. It will still form tracts there. The decassation will take place on the second order neuron, and then it will reach the thalamus again. And so even the second order neuron here will also form another tract, just like the first order neuron also forming another tract. The third order neuron terminates at the level of the cerebral cortex, the region of perception, which again is postcentral gyrus. So the concept here is that we need to know that there are three levels of neurons all the way to the cortex, first order, second order, and third order neuron. At the cortex, the cortical region where the third order neuron terminates will depend on the sensation coming in. For example, the sensations which are going to the postcentral gyrus are the ones which are for general sensation. But we know from our neuroanatomy, neurophysiology lectures that we have different cortical regions for different sensations, and especially the special sensations that we're going to look at today. Remember, as the axons of first and second order neuron traverse the brainstem and spinal cord, they form ascending tracts. And the image on your left captures the different tracts of the spinal cord. I hope you do remember them for general sensations. So those ones were general sensations. I'm bringing this up so that you know that there are some things which are basically a common template, but we've reviewed that enough. So I want us to focus on the first two special sensations. I want us to focus on the olfactory sensation and gustatory sensation in this first part of the lecture. Let's begin with the olfactory sensation. So we're going to look at the structure of the nasal cavity. We'll also look at the structure of the olfactory epithelium and we'll describe the pathway of smell. This is how the nasal cavity looks like if you are to cut in coronal section. If you take a coronal section of the nose, we'll see something like this. Usually, the nasal cavity consists of two nasal airways, the right and the left side. Well, evident by when you look at the anterior part of your nose, you'll see two openings. Those two openings are the ones we call the nostrils, and they lead you to each of the nasal airway. There's a septum that divide the right and the left nasal airway, so that we have the nasal septum there. That septum can either have cartilaginous parts and bony parts. So it is both cartilaginous and bony septum. Of course, it's lined by mucosa either way. The lateral wall of the nose has these projections. There are still bony projections. We call them tabinates or conchae. The purpose of these projections is to increase the surface area over which the mucosa of the nasal epithelium will lie. And so this is the superior nasal tabinate, this is the middle nasal tabinate, and that's the inferior nasal tabinate. We have three of them. The grooves between the tabinates are the ones we call the meatai. So this is the inferior nasal meatus, this is the middle nasal meatus, and that's the superior nasal meatus. Remember, this is the roof of the nose, the upper part, and this is the lower part. So this is the palate here. The oral cavity will be below this point. Now, the cavity of the nose, whether which chamber, is lined by mucosa. And based on how that mucosa looks like, based on how that epithelium looks like, we can describe three histological regions of the nose. We have a region we call the nasal vestibule. We have a region that we call the respiratory region, and we have a region that we are terming the olfactory region. So the nasal vestibule is this region here, just behind the nostril. The respiratory region is this region here, and the olfactory region is that region there. Let's look at them one by one. We start with the nasal region, the nasal vestibule, or rather. So the nasal vestibule is the region of the nasal cavity just behind your nostrils. That region of the nasal cavity just behind your nostril is lined by epithelium similar to that one of the skin. The skin has what we call stratified squamous keratinized epithelium. When you say epithelium is keratinized, it means that it has several layers of cells. When you say epithelium is squamous, it means that the cells on top are flattened. So stratified squamous epithelium is what the type of epithelium we usually find in regions that are more subject to friction, like the skin. If there's a lot of friction being experienced on such surfaces, usually it has a layer of keratin as well, as we see in the skin, and that extends also into the nasal vestibule. So the epithelial lining of the skin and that of the nasal vestibule is similar. This is how that epithelium would look like. That region is the region of the epithelium. It's a lower magnification, but we can see some keratin and we can see the epithelium. This is connective tissue corresponding with the dermis of the skin, basically. So in simple terms, the nasal vestibule is lined by skin. That skin has a lot of hair, and that's what you're seeing here. These are hair follicles as they're within the dermis. It contains a lot of hair. The hair of the nose is what we call the vibrissae. Other than the hair, usually hair also usually associated with sebaceous glands, and so there are also several sebaceous glands in that region. We're just saying it's a hairy skin. Now, important to note is that that region of the nose does not contain the receptors for smell. It doesn't. So that is it, the nasal vestibule. The second part is the respiratory region. The respiratory region refers to the lower 2/3 of the rest of the nasal chamber if you remove the vestibule. The lower 2/3 of the rest of the nasal chambers. This region of the nose is lined by a different type of mucosa. It is lined by the respiratory mucosa. We define respiratory mucosa as pseudostratified columnar ciliated epithelium with goblet cells, and beneath it is usually what we call the lamina propria. So let me say that slowly again. When we say epithelium is pseudostratified, it means that you basically have a mixture of short and tall cells. And so histologically, it will look like some nuclei are at different levels. So some nuclei are on the basal side and others somewhere. And generally, the top of the epithelium is usually lacking in the nuclei. That's how pseudostratified columnar epithelium looks like. So this one, that epithelium is ciliated, and that's the ciliary lining. We can see that one there. And it usually has a lot of goblet cells. Goblet cells are mucus cells. So that's how the epithelium looks like. Or in this image, we can capture that. You see it's a mixture of short and tall cells. Beneath the epithelial lining is a loose connective tissue layer, which we usually call the lamina propria. So when you talk of respiratory mucosa, we are referring to the epithelial lining and its lamina propria. This is the epithelial lining, and this is the lamina propria. So this mucosa is the one that adheres onto the bones of the nose. It's the one that will adhere to the different bones of the nose. It's the one that line the respiratory region of the nasal chamber. Important to note is that this epithelium does not have, again, the olfactory receptors, but instead it has several mucus cells. Maybe it's important to remind ourselves the cell types that are usually found on this epithelium. This is how the respiratory epithelium look like at a lower magnification or relatively lower. So the ones you're seeing here, large non-staining parts, are the mucus cells. The cell types of the respiratory mucosa are the following. The most abundant are the ciliated columnar cells. These are the cells that contain cilia. They're the ones that form the protective barrier. But as we know, other than forming the protective barrier, the cilia they contain help to propel substances on the surface of the epithelium. Goblet cells are these ones which produce mucus. So the mucus produced by the goblet cells trap particles in the nose. And then that mucus is propelled by the cilia to a particular direction, usually posteriorly. That is what we call the mucociliary rejection current. So the ciliated columnar cells and the goblet cells work in collaboration. That collaboration is termed the mucociliary rejection current that help to trap and extrude substances away from the respiratory tree. The third cell type are the basal cells. They're usually the short ones. These ones are stem cells. Basically, they give rise to the other cell types. We have the small granule cells. You can call them small granule cells or the Kulczycki cells. These cells produce hormones. They are part of what we call the diffuse neuroendocrine system. Cells which produce hormones in response to neuronal stimuli. diffuse neuroendocrine system. Lastly, we have the brush cells. The brush cells are chemoreceptor cells. They detect concentration of oxygen, perhaps, within the respiratory tree. So these ones are the cell types of the respiratory region. Important to note, the respiratory region has a lot of mucous cells and does not have the olfactory receptor cells. The last and perhaps the most important one for this lecture is the olfactory region, which is now the upper 1/3 of the nasal chamber there. So the olfactory region is lined by the olfactory mucosa as opposed to respiratory mucosa. Once again, when you talk of olfactory mucosa, we are referring to a pseudostratified columnar ciliated epithelium, but now without goblet cells. It's a pseudostratified columnar ciliated epithelium that does not have goblet cells. Deep to that epithelium is a lamina propria. Now, what's unique about the lamina propria for olfactory epithelium? The lamina propria is still of loose connective tissue, true, but there's something else. That lamina propria contains several glands, like that one or those ones. These glands are known as the Bowman's glands. In addition to those glands, the lamina propria of olfactory mucosa also contain nerve bundles, like this large thing is a nerve bundle. So these nerve bundles are, of course, the nerve bundles of olfactory nerves. The olfactory region of the nasal cavity is lined by olfactory mucosa, which has that epithelium and that kind of lamina propria. So this image captures for you how the olfactory epithelium would look like and how the lamina propria for the olfactory epithelium look like. Importantly, we have the Bowman's glands in the lamina propria, and we have nerve bundles in the lamina propria. In the epithelium itself, it's a pseudostratified columnar ciliated epithelium that does not have goblet cells. And you can see the pseudostratification here. All the cells reach the base, but we have a mixture of short and tall cells. Now, there are 3 major cell types on the olfactory epithelium. The olfactory receptor cells, which is this one or that one. The olfactory receptor cells are basically chemoreceptor cells. They detect chemicals. As we can see in this image, these olfactory receptor cells are actually bipolar neurons. Because you can see this is the cell body. The axon goes to form the optic nerve. The dendrite is on this side, on the surface, with those ciliary processes. So the cilia of the olfactory mucosa come from the bipolar neurons of the olfactory epithelium, rather than from the supporting cells, as you may have expected in the respiratory mucosa. So these ciliary extensions are basically the dendritic processes of the bipolar neurons, which we call the olfactory receptor cells, and they are chemoreceptor cells. They detect chemicals. So what usually happens is that the Bowman's glands will produce some secretions that can dissolve the chemicals that you're breathing in, and then that will then be detected by the cilia of the olfactory receptor cells. The second cell type are the sustentacular cells. We call them sustentacular or supporting cells. So they provide structural support to the epithelium. As we can see, the sustentacular cells of the olfactory mucosa may have microvilli to help in absorbing the secretions which have been released, so that we don't have to have a running nose all the time. The basal cells are these ones, similar to the basal cells of the respiratory epithelium. these cells are basically stem cells. So we've talked about the nasal cavity. Importantly, two types of epithelia come out. On your left, olfactory epithelium, and on your right, respiratory epithelium. Very different types of epithelia that line the nasal cavity. Now we can talk about olfactory relay pathway. So olfaction, the sense of smell, is detected by the chemoreceptor cells of the olfactory epithelium. As we've seen, the olfactory epithelium occupies the upper 1/3 of the nasal cavity. Now, in particular, that epithelium takes or occupies about 10 centimeter square area of the nasal cavity on the upper part. The type of structures it actually line, the olfactory mucosa will line the roof of the nasal cavity, which is occupied by the cribriform plate of the ethmoid bone. It also lines the upper part of the nasal septum, and it lines the superior nasal turbinate as well. So that is the region occupied by the olfactory mucosa. That's where the olfactory mucosa line. The superior nasal concha, the upper part of the nasal septum, and the cribriform plate of the ethmoid, which basically the roof of the nasal cavity. So once the sense of smell has been detected by the chemoreceptor cells, which are bipolar neurons, those neurons go through the cribriform plate of ethmoid bone. Remember, they are first-order neurons. The olfactory nervelets are basically first-order neurons. These first-order neurons then go through the cribriform plate of the ethmoid bone to find themselves into the anterior cranial fossa, where the synapse now in that region, which we call the olfactory bulb. The olfactory bulb contains the cell bodies of the second-order neurons for the pathway of smell. So those neurons whose cell bodies are within the olfactory bulb will have their axons leave the olfactory bulb in this thing we call the olfactory tract. Great. Now, if you remember, I told you that most sensations will take the template of first order, second order, and third order neurons. That applies for all sensations in the body except olfaction. Olfaction doesn't have the 3 orders of neurons before it reaches the cortex. It only has 2 orders of neurons, then it will reach the cortex. And so the olfactory bulb contains cell bodies of olfactory second-order neurons for olfaction will give rise to the olfactory tract. And these olfactory tract fibers are the ones that terminate in the cerebral cortex. So for olfaction, the second-order neuron reaches the cerebral cortex. In this image, you can see this is the location expected to be for the olfactory bulb, and then this one for the olfactory tract. So this olfactory tract usually projects to the cerebral cortex in the region we call the primary olfactory area, which is in the piriform lobe. So if you remember, piriform lobe consists of 3 things. We have the uncus, area number 34. We have the entorhinal cortex, area number 28. And we have the lateral olfactory stria. So uncus, entorhinal cortex, and the lateral olfactory stria. Now, the olfactory stria are basically divisions of the olfactory tract. As you can see, this one coming here, that's called the medial olfactory stria, and that is the one that decussates. Remember, second-order neuron decussates. And then this other green part there is the lateral olfactory stria. So that lateral olfactory stria, the uncus and the entorhinal cortex constitute the primary olfactory area, what we call the piriform lobe. And that is where the sense of smell is perceived. So that is the pathway of olfaction. I want to now talk about the pathway of taste. So for the pathway of taste, we'll first talk about the anatomy of the oral cavity. We'll then talk about the general structure of the tongue and the epithelium that lines the tongue. Then we'll talk about the taste buds, and we'll talk about the pathway of taste sensation. Let's start with the general anatomy of the oral cavity. So the oral cavity has two major compartments. There's this region here that is containing the tongue and is limited by the teeth. That is what we call the oral cavity proper. But there's also this region here between the teeth and the cheeks or the lips. That is what we call the vestibule of the mouth. We can see that the oral cavity proper and the vestibule of the mouth is separated by the teeth. So this, if you look at the teeth from up downwards, that axis, we have the teeth either up or down, then we have the gum either up or down, and then we have the jaw, what we call the alveolar processes. The alveolar processes, the gum and the teeth constitute the dental arch. So from above, you'll have the alveolar processes, then gum, then teeth, then gum, then alveolar processes. That is what we call the dental arch. So internal to the dental arch is the oral cavity proper, and external to the dental arch is the vestibule of the mouth. The palate forms the roof of the oral cavity proper. The palate has two parts. The anterior part of the palate is osseous. It has bone. And so we call the hard palate. The posterior part has soft palate. The posterior projection of the soft palate is what you call the uvula. We know that the mouth is continuous with the throat. The throat is what you call the pharynx. The mouth is continuous with your throat. The junction between the mouth and the throat is demarcated by this fold there, which we call the pillars of Fauces. So that fold there, just in front of the palatine tonsil, or that one, just in front of the palatine tonsil, is what we call the pillars of Fauces. Maybe a better image for that would be this one. This is the pillars of Fauces, which distinguish the junction between the oral cavity proper and the pharynx. Now, the reason we're discussing oral cavity is because we want to discuss taste. The receptors for taste are located on the tongue, most of them there, but they're not just on the tongue only. They're also found on the palate, a few of them, and also on the epiglottis, again, a few of them, but most of them are found on the tongue. Having said so, we can now describe the anatomy of the tongue. So the tongue is a muscular organ made up of muscles, muscles inside it and muscles external to it. The muscles which come from outside that go to it are called extrinsic muscles of the tongue. The muscles which are within it are called the intrinsic muscles of the tongue. So it's a muscular organ, highly muscular, then lined by the mucosa of the tongue. This muscular organ is for swallowing, but we also know it for taste, and also for speech, among the primary functions of the tongue that we might be comfortable mentioning at this point. Now, there's a part of the tongue that is in the oral cavity, and there's a part of the tongue that is in the pharynx. So the anterior part of the tongue, the anterior 2/3 of the tongue is in the oral cavity, in the mouth. The posterior 1/3 of the tongue is in the pharynx. The tongue has a root. The root is this region here that it's using for attachment to the hyoid bone and to the mandible, this region here, that's the root of the tongue. So that root is attaching to the mandible, to the hyoid bone, and even to the pharynx actually. It has an apex, which is that one. You may call that the tip of the tongue. It has a dorsum, this is the dorsum of the tongue. And as you can see, for the anterior tooth of the tongue, the dorsum faces up. And for the posterior 1/3 of the tongue, the dorsum faces posteriorly. That dorsum is convex, as you can see, on all sides. Then you have the inferior surface of the tongue, this one, which you also call the ventral surface of the tongue. The tongue is attached to other things through its extrinsic musculature. So there are muscles which attach the tongue to the palate, to the mandible, the styloid, and also to the hyoid bone. So the muscles will have different names depending on where they're attaching to. We don't have to go to the names, the individual names of the extrinsic muscles of the tongue really, but understand that there are muscles which will hold the tongue to those external structures. We call them the extrinsic muscle of the tongue. Maybe the only one that is lacking muscle direct is the attachment to the pharynx. Grossly, the tongue has a V-shaped groove on its top. That V-shaped groove here is called the sulcus terminalis. This sulcus terminalis is actually the one that vaguely divides the tongue into two parts. The anterior 2/3 of the tongue, which is now found within the mouth, therefore called the oral tongue. You can call the presulcal tongue. If this is the sulcus, then this is presulcal. And we have the posterior 1/3 of the tongue, which is now found in the pharynx, and so it's called the pharyngeal tongue or the postsulcal tongue. It is V-shaped, it's an inverted V, as you can see. The center of the V is a small depression called foramen cecum. This foramen cecum marks the embryological site where the thyroid gland began its development before it migrated to where we know it is found right now. But originally, in the embryo, the thyroid gland begins from there. So that depression that left marks the path that was followed by the thyroid gland as it left the oral cavity. Anyway, the mucosa of the tongue is usually pink and moist most of the time and is adherent to the underlying intrinsic musculature of the tongue. The intrinsic muscles of the tongue are the muscles within the tongue. They don't live, they're just within the tongue. Some run longitudinally, some vertically, some transversely. So when you talk about that mucosa, we are referring again to the epithelial lining and its lamina propria. Now, importantly, the mucosa on the dorsal side of the tongue has several projections. Those projections are called papillae. Those projections, which are called papillae, are the ones that house the receptors for taste, which we call the taste buds. So in this image, we can see several types of papillae of the tongue. We have different types of lingual papillae. The most numerous of them all that you'll see in your neighbor's tongue or in your tongue, and look at it in a mirror, are the ones we call the filiform papillae. They are very tiny, several all over the tongue. Filiform papillae. Then we have a few within the anterior 2/3 of the tongue that appear slightly bigger. than the filiform. So those ones which come out slightly bigger than the others are the ones we call fungiform papilla. They look like the top of a mushroom that has not yet everted itself. The third type are the ones we call the foliate papillae. They're on either sides of the tongue, not so well developed in humans. Hard to see them, but when you see one, they appear like lines. The largest of them all are the circumvallate papillae of the tongue. The circumvallate papillae of the tongue are largely found in the posterior aspects of the tongue, but they're the largest. When you see one, you'll think it's a pimple or a boil. They're the largest papillae. So this is a cross-sectional image of one of the papillae of the tongue, in this case, circumvallate papillae. And we see that projection there. Adjacent to it are some grooves. So this is how the tongue epithelium looks like. First of all, look at that epithelium. It is stratified squamous, parakeratinized epithelium. Parakeratinized because there's a thin layer of keratin on the surface. Deep to it is this connective tissue zone that contains several glands. These glands which are contained within the tongue are called the glands of von Ebner. These glands also produce some secretions that dissolve substances in the tongue that need to be tasted. The glands of von Ebner are deep on the connective tissue part of the tongue. The epithelium of the tongue is stratified squamous parakeratinized epithelium. This other image captures that, although this one doesn't look parakeratinized, this one looks non-keratinized, but that's the whole concept. Parakeratinized epithelium has patchy keratin. So some regions will not have the keratin lining. So this is a higher magnification of perhaps a zone here, so that we see these tiny things at better view, like this. This space here, that groove is that one there, the pericircumvallate groove in this case. So these rounded structures, ovoid structures, which seem to be interrupting the epithelium, are the ones we call the taste buds. They're usually found in the grooves of the papillae. They're on the epithelium, but adjacent to the groove of the lingual papillae. The taste buds are the receptors for taste. So one would look like that, with the surface there being an open space. That's the taste pore, and looking around like that. There are some cell types of the taste buds that I want us to talk about. We have the taste receptor cells. You can call them gustatory cells. So the gustatory cells are the taste receptor cells. These taste receptor cells are chemoreceptor cells. They detect chemicals within the epithelium of the mouth. Other than that, we also have the sustentacular cells, which are basically supporting cells. They provide structural support to the taste buds. And lastly, we have the basal cells. As you mentioned, even for olfactory epithelium, the basal cells here are for regeneration. They give rise to the other cell types. So those are the three types of cells which we find in the taste buds. What kind of taste sensations are we able to pick? We're able to pick sweet taste, sour taste, bitter taste, salty taste, and umami taste. Umami taste is the taste of meat. So it's a meaty taste, largely because of some amino acids like glutamate being present. There's a concept of taste map where we talk about some regions of the tongue having a threshold for some particular taste sensations. That is true, but not fully. What I mean is this, that even though sweet taste can be felt all over the tongue, the region for highest predilection for sweet sensation, taste is the anterior two-thirds of the tongue. So this is where we get sweet taste sensation. Salty taste is largely on the anterolateral aspects of the tongue, sour taste on the lateral aspects of the tongue, bitter taste usually posteriorly, and umami perhaps on the dorsum of the tongue. But remember, you can feel any of these tastes anywhere on the tongue, but those regions are the ones which will give us the best results. Having said so, let's finish this first part of the lecture by talking about the pathway of taste sensation. So once taste has been detected by the taste buds, and in particular, the chemoreceptor cells within the taste buds, we've called them the gustatory receptor cells. That information is relayed through first order neurons that carry taste. The first order neurons that carry taste are many depending on the region they come from. The taste buds in the anterior two-thirds of the tongue will relay their sensations through the seventh cranial nerve, which we call facial nerve. The specific branch of facial nerve that run to the tongue for taste sensation is called the coda tympani nerve. The cell bodies of those neurons will be within a ganglion within the facial nerve found somewhere in the ear, which we call the geniculate ganglion. The taste buds from the posterior one-third of the tongue are relayed by glossopharyngeal nerve. So the cell bodies of those neurons will be in the glossopharyngeal ganglion. Similarly, the taste buds from the sensation of taste from the taste buds on the epiglottis are carried by vagus nerve, the 10th cranial nerve, and they'll also have their own cell body location, a ganglion somewhere. Whichever the taste has come from, the information carried by the first order sensory neuron for taste goes to the brainstem. It will terminate in a nucleus within the brainstem. Remember, a nucleus is a collection of cell bodies of neurons within the white matter of the CNS. So that will be containing cell bodies of the second order sensory neurons. We call that nucleus, the gustatory nucleus. It is found within the brainstem. From the gustatory nucleus, we have second order sensory neurons for taste. They'll decussate and climb all the way to the thalamus, as we said. So after decussation, they go to the thalamus. From the thalamus, we'll have synapse in the third order neurons, which will take information from the thalamus to the cortical region for perception of taste, what we call the primary gustatory cortex, Brodmann's area number 43, which is found somewhere in the parietal operculum. If you remember the operculum, so the parietal operculum, that's where you find the primary gustatory cortex, just next to the insular region. And that's why sometimes you say the parainsular cortex. So that is the pathway of taste. And that summarizes for us what I wanted us to capture in the first part of the lecture, focusing on some reviews that we just said. Then in particular, we've looked at olfactory sensation and taste sensation.